All right, in this video, we're going to talk about the details of what's happening at a synapse. So in our previous videos covering the nervous system, which you can find before this one in the playlist, we talked about resting membrane potential, how we generate graded potentials to hit threshold and get an action potential, right? And so now we're going to zoom in on the details of how exactly neurotransmitters are released at synapses and how they produced graded potentials inside of cells. So in this zoomed out view, you can imagine starting at the very beginning of this, we had an action potential that would be zooming down this axon. It's myelinated, so we should draw it as hopping from node to node. Remember your nodes of round VA. And that action potential is going to then hit all of these axon terminals, right? Each of those axon terminals is going to form a synapse with this postsynaptic neuron. If enough of those signals are excitatory, remember EPSPs, excitatory postsynaptic potentials, then we'll hit threshold right at that axon hillock. If you hit threshold, now we get another axon that's going to send an action potential right further on down the line. And so all we're really doing at this point is zooming in very closely on one of those synapses and asking, how does the release of neurotransmitters actually cause a graded potential? All right, so this picture is a good starting point, but we have a handful of parts that we need to fill in. So the first point to make is this all starts with an action potential coming down this axon, right? And remember that an action potential is just a wave of voltage. When we draw it on a graph and follow it over time, the y-axis, we're measuring millivolts, right? So if this is our threshold line, we had excitatory postsynaptic potentials back in this neuron cell body up above the screen where we can't really see that hit threshold, and our action potential looks like this. So it's just a big wave of voltage. Once you get to this axon terminal, these channels that they're showing all over the axon terminal, those are voltage gated channels, but now instead of sodium, these are voltage gated calcium channels, right? Calcium tends to accumulate outside of cells, outside the cell membrane. And so when you open them, we expect calcium to flow into this cell, right? And the voltage gated part, remember, just tells you that the channels are gated, they can open and close, and the thing that opens the gate is voltage. So remember, what is an action potential? It's a big wave of voltage. So the action potential coming down the axon will open those channels, calcium will flow in, and there are a lot of molecular details going along here, but we probably don't need to know every one of them. It's probably enough just to know that calcium inside this axon terminal is the trigger that tells these vesicles, right? These round structures, they're really like spherical balls of membrane that are filled with neurotransmitter, and the little green dots are representing our neurotransmitter. So when you have calcium enter the axon terminal, you expect those vesicles to fuse with the membrane of the axon terminal and spill their neurotransmitters out into the synaptic cleft, right? That term synaptic cleft really just means the very, very tiny space in between the two neurons, right? Remember, in all of this, we call this guy the presynaptic neuron because it's sending information into the synapse, and this is the postsynaptic neuron because it's on the receiving end of the synapse. All right, so at this point, we have had our action potential. We have calcium that's entered the axon terminal. That's triggered the release of our neurotransmitter. And nothing's going to happen unless this neurotransmitter happens to bind and match with a receptor on the postsynaptic membrane, right? So those receptors, they're showing these as ligand-gated ion channels. They also could be metabotropic neurotransmitter receptors. But either way, the end result is ion channels open. And so in this example of these being ligand-gated ion channels, let's pretend for a second that this is a ligand-gated sodium channel. What that means is that when our neurotransmitter binds to our receptor, we expect it to open, and they're not really showing us sodium here. But remember, sodium is built up around the exterior of all membranes at all times. And so if we open a sodium channel, we expect it to diffuse from high to low concentration into the cell. Okay, now, if you remember on our action potential graph, right, this is threshold. Remember, our negative 70 resting membrane potential value is below that yet. This was negative 55 at threshold. We start off at rest. If you open this voltage-gated sodium channel, ligand-gated sodium channel with your neurotransmitter, all that means is you're letting sodium into the cell. The graded potential is going to rise, right? That is our graded potential. Specifically, it's excitatory because we're going up on the graph. So EPSP, excitatory postsynaptic potential. If this had been a potassium or chloride channel, it would have made our graded potential go down and hyperpolarize, and that would have been an inhibitory postsynaptic potential. All right, so if we open enough of these and we let a lot of sodium come in, and usually this has to happen at multiple synapses, right? One synapse on a neuron is not enough to make it hit threshold. But if this happens over and over and over again, you can drive your potential hard enough upward to cross threshold, get an action potential. And at this point, remember, context, that action potential is now going to be happening in the axon hillock of the same neuron that was the postsynaptic neuron in our diagram. And so it's really just this chain of repeating action potential, neurotransmitters, action potential, neurotransmitters. And that will keep happening as long as you keep reaching threshold, right? If there was a neuron where there just wasn't enough excitatory postsynaptic potential created and you never hit threshold, then none of this would happen, right? If we failed to hit threshold and then the neurotransmitter was removed, you would just come right back down to resting and nothing would happen at all. That's the idea that we went over in the action potential video, that action potentials are all or nothing. They either happen or they don't. And so there's no such thing as half of an action potential because you either crossed that threshold value or you didn't. All right, so now we can ask the question, what's going to happen to those neurotransmitters after they're released? You've got a few different options. One is you can just let them diffuse away, right? So if you give neurotransmitters enough time, just like any molecule, they'll diffuse and sort of scatter away from the synaptic cleft, which means they're leaving where the receptors are and they won't have an effect anymore. Option two, you can have recycling back into the axon terminal. In that case, you would find active transport pumps or channels like this guy in the axon terminal membrane, and they would take the neurotransmitters and pump them back up in to get reused a second time, and a third time, and a fourth time. Since you're removing them from the synaptic cleft, recycling is another way to get rid of the neurotransmitter and prevent it from being taken back up. There are a handful of drugs that work this way. If you've ever heard of a category of drugs called SSRIs, selective serotonin reuptake inhibitors, those are medications that block those channels and prevent the reuptake of serotonin, which means the serotonin stays trapped in the synaptic cleft and has more of a response. So I know it's called an inhibitor, but if you block reuptake, that would actually make the neurotransmitter more active by leaving it in the synapse for longer. And those drugs can be used to treat depression because serotonin tends to make people feel good. And so if we have more serotonin in the synapse, you get more of that effect. Okay. The third option is you can use an enzyme to intentionally degrade and destroy the neurotransmitter. In that case, you would be putting your enzyme in the synaptic cleft itself as a way to very rapidly get rid of the neurotransmitter. So if you imagine that these little guys are enzymes, they would be metabolizing and destroying that neurotransmitter to get rid of it. Enzymes can work unbelievably fast. And so the only reason that you would do this is if you need speed, right? It's easy to think that releasing the neurotransmitter quickly is the critical part of this. But remember that getting rid of the neurotransmitter quickly is equally important. if you have something like a muscle contraction going on and your brain says it's time to relax, if you can't get rid of the neurotransmitter, the muscle won't relax and it would just stay contracted and locked down until this like slower process of diffusion happened. And so whenever speed is very, very critical, we'll often use those enzymes to get rid of the neurotransmitter at a much, much faster rate. All right, so let's do a summary, put some things together. If you want to know what effect a neurotransmitter is going to have on the postsynaptic cell that it's being released on, we have a handful of questions to ask. One is, which ion is being manipulated? That just means which ion flows through the channel that ends up being opened. If you open sodium channels, right, increasing sodium permeability, sodium has a positive charge and it enters cells, right? So if this is our cell membrane, since sodium diffuses in, it causes an increase in charge, that's depolarization, and we say that's excitatory. Excitatory, same thing as saying it causes EPSPs. If you get permeability of either potassium or chloride, that causes an inhibitory response, IPSPs. And that will cause repolarization, or if you go below resting, we'll call it hyperpolarization. Remember, the reason for that is potassium builds up inside of the cell, so if you let it diffuse, it moves out. In that case, you're losing a positive. Right? Losing a positive makes you become more negative. Chloride is built up outside of cells and has a negative charge, so it tends to flow in. That means you're gaining a negative. So whether you're losing a positive or gaining a negative, those things will make you inhibitory. If you're gaining a positive, like sodium, that would be excitatory. All right, so once you know which ion is moving, you at least know generally what the effect is going to be, EPSP or IPSP. From there, you can ask, all right, how much of the neurotransmitter is released? There are a lot of variables that come into this, but the biggest one is action potential frequency. Right? Every time an action potential comes down this axon terminal, it's going to cause those voltage-gated calcium channels to open, which is going to trigger neurotransmitter release. So if you pulse more action potentials down the axon, you get more calcium. More calcium means release more neurotransmitter. Okay, so that's the how much question. And then lastly, duration. How long does the neurotransmitter stay in the synapse? That's what we just talked about with diffusion, with reuptake. Enzymatic degradation would also fit into that category. The longer the neurotransmitter stays in the synapse, the more effect it's going to have. All right, so now tying everything together as much as possible. When we're trying to decide whether an action potential is going to happen and whether a neuron is going to hit threshold, this concept is called summation. I mentioned it briefly in one of the previous videos, but the idea is you're adding up the effect of all the synapses on a cell. All right, so remember, when you see a neuron like this, it's not just one synapse. They're showing you one neuron coming in here, but all of these dendrites would also have synapses connecting to them as well. And so if we add them all up, there might be 100 synapses over the course of the cell. Some of them are excitatory, some of them are inhibitory. Summation really just means you're adding up all the excitatories and inhibitories, and after they cancel each other out, what's left over? If the net effect is more excitation, then the cell's excited. If the net effect is inhibition, then it's suppressed and you don't hit threshold. So temporal summation, tempo, just like time in music, like keeping time, tempo of the beat. Temporal just means you're adding up the effect of synapses that are firing over time. Spatial summation means over three-dimensional space, right? So spatial summation would be multiple synapses on the same neuron firing together. Temporal would be those synapses firing repeatedly over time. This is all just a very fancy way of saying you add up the effect of all the synapses on a neuron over their space and over time. And whether you hit threshold or not, that's the only question that matters. And so whether it's more of a temporal thing, repeated firing of a small number of synapses, or spatial, right, a bunch of synapses firing all at once, either way, you hit threshold, the action potential will be the same regardless. All right. Okay, so hopefully this was helpful in tying everything together. This is kind of the end of the action potential story. The next video that I do is going to get into how neurotransmitters are regulated a bit more carefully, how pharmaceuticals and drugs come into play, so we can use those molecules to sort of mimic or inhibit the way that naturally occurring neurotransmitters affect the receptors. All right, so as always, make sure you follow along in the playlist so that you see these videos in order, and subscribe to the channel so that you get any new videos that pop up, because I'm making these consistently. All right, check it out next time for drugs, agonists, antagonists, and how they impact neurotransmitter receptors.